Yes.
Perfect. Well, first of all, I would like to thank the organizers for inviting me to be here today. It's a really a pleasure, and all the best with this seminar series. I think that's a fantastic idea to have this series of seminars talking about urban ecology. Today, I'm going to talk to you about some of the research that we conduct in the Biofarm Lab about soil biodiversity and ecosystem function, and of course about urban ecosystems. But first of all, I would like to start by stating the obvious. And the obvious is that soils that are very important for human well-being. 95% of the food that we consume depends directly or indirectly on our soils. And however, a third of our soils at a global scale, they're already highly degraded. Only in the European Union, 6 out of 10 soils are in an unhealthy situation, according to the European Commission. And the problem is that soils are a non-renewable resource, and a centimeter of soil can take thousands of years to develop. And because of that, it is essential that we learn how to protect, how to conserve our soils for the next generation. And in my opinion, in order to protect our soil, the first step is understanding the complexity of soil biodiversity. Soils are the largest reservoirs of biodiversity of the planet. 6 out of 10 species, they depend directly or indirectly on our soils. This includes, for example, vascular plants, which roots they go through the soils, non-vascular organisms such as biograss-forming mosses, and also a myriad of soil organisms such as bacteria, fungi like mycorrhiza, protists, or invertebrates like some invertebrates like nematodes. They are the most abundant animals of the planet. Visualizing the complexity of the soil microbiome is very complicated, and because of that, I brought you this very simple example. The soil that fits in a teaspoon already contains thousands of species of bacteria and millions of individuals. This teaspoon of soil already contains more individuals of bacteria than millions of inhabitants live on planet Earth. So these soil organisms, they are the unseen majority that also conduct almost every single ecosystem function. And that includes, of course, carbon sequestration, organic matter decomposition, primary productivity, nutrient cycling, soil stability, and a bunch of different soil processes, but also they are critical for every single aspect of our life. Soil organisms are essential for one health, the health of plants, animals, and the environment, and soil organisms are essential for every single aspect of our life, including some aspects that we don't think about them very often, like, for example, the conservation of monuments. And I would like to show you some examples from our own research. The first example is coming from a decade of research where we have been investigating the relationship between soil biodiversity and ecosystem multifunctionality, the capacity of an ecosystem to support multiple functions simultaneously at the global scale, at the regional scale, within particular biomes such as dryland, also outside dryland ecosystems, and in mesocosm, microcosm experiment, where we have been finding systematically that soils that have a greater biodiversity of bacteria, fungi, protists, and invertebrates, they also have greater ecosystem multifunctionality. And that means more carbon sequestration, more organic matter decomposition, more soil fertility, productivity, pathogen control, etc., etc. Of course, this is also the case in urban green spaces. At the global scale, using a network of collaboration that we established almost 10 years ago, we investigated the relationship between soil biodiversity and multifunctionality across city parks and gardens, and we found exactly the same. Soils that have a greater biodiversity, city parks and gardens that have soil with greater biodiversity, they also have greater ecosystem multifunctionality, meaning better nutrient cycling, water regulation, carbon sequestration, plant productivity, etc., etc., meaning ecosystems that they are more easy to manage from an economic point of view. So soil biodiversity is the unseen majority in our parks, and they are working for us to support ecosystem function. Soil biodiversity is also essential for supporting the stability of terrestrial ecosystem, the temporal stability of ecosystem productivity. And our own research have provided evidences across global surveys that soils that have a greater biodiversity of beneficial organisms, such as, for example, decomposers, fungal decomposers, they also have greater ecosystem stability. However, soils that have a greater number of species of plant pathogens, unfortunately, they have lower ecosystem stability. So With these three examples, I just wanted to show you that soil biodiversity is critical for ecosystem function, especially very important for soil processes, but I would not like to leave you with the idea that soil biodiversity is only important for soil processes. Soil biodiversity is critical for every single aspect of our life, and that includes, of course, one health, the health of plants, animals, and the environment, which is so critical in urban ecosystems. Not sure you're aware of this, but soils are the largest reservoir of plant pathogens of the planet. Plant pathogens that they can have an impact on our capacity to produce food and therefore in food security. An emblematic example of this is the case of Phytophthora infectans that caused the great Irish potato famine in the 19th century, killing 1 million of people out of famine. The population of the island was reduced by 25% of the population. So these proteozoa mycota have a huge impact on our capacity to produce food and our own health. And because of that, one of the main goals of my lab is to produce innovative knowledge, tools that can be used by policymakers, land managers to take informed decisions about these important organisms. Here in this slide, for example, you have a global map with the distribution of the genus Phytophthora in soils under current conditions, but we are also very interested in working under global change scenarios. Unfortunately, soils, they are not only the largest reservoir of plant pathogens of the planet, they are also one of the major reservoirs of human pathogens and antibiotic-resistant genes. Pathogens such as, for example, Clostridium tetanus, Yersinia pestis, and also less described pathogens such as fungal pathogens, in this case of Aspergillus, they are very prevalent in our soil. Aspergillus, for example, can cause aspergillosis with 2 million infections per year, highly fatal if you have an immune-depressed system. But also soils are one of the major reservoirs of antibiotic-resistant genes. I'm not sure if you're aware, but only in the European Union, 35,000 people die every year out of antibiotic resistance. Antibiotic-resistant genes, they are very important, especially coming from the environment, understanding their distribution, because many times they can jump to clinic pathogens. So understanding the distribution of these organisms is very important for our own health. And of course, soils in urban green spaces, in city parks and gardens, they are not different to other soils in that way. Soils in urban green spaces, they have a huge amount of microorganisms and also of pathogens. Aiming to understand how many organisms of this type, what type of organisms live in our urban green spaces. Almost a decade ago, we put together this global collaborative network where we investigated the influence of urban green spaces, city parks and gardens compared to nearby natural ecosystems. And what we found was something pretty straightforward. Urban green spaces, they have a larger prevalence of plant pathogens compared to natural ecosystems and a lower prevalence of beneficial organisms such as mycorrhiza fungi. Also, urban city parks and gardens, they have a greater prevalence of certain groups of antibiotic-resistant genes, viruses, and also some bacterial taxa like mycobacterium that can cause respiratory disease. So very important that we understand the distribution of these organisms. Fungal communities are not different that way. We also found that some fungal human pathogens, like for example, Trichosporum, that can affect humans with an immune-depressed system, they are also very prevalent in city parks and gardens at the global scale, but also in an intensive survey that we conducted in Shanghai. So very, very important to understand the distribution of these organisms, what microorganisms live in our city parks and gardens for our own health. In this particular context, soil biodiversity is also very important to filter the entrance of pathogens. Our own research have provided evidences at the global scale across natural ecosystems that soils that have a greater native soil biodiversity, they also have lower prevalence of pathogens. In the case of city parks and gardens, this is also something that we found, but in this case, it looked like the diversity of invertebrates, soil invertebrates, is very important to regulate the entrance of pathogens. Of course, pathogens, plant pathogens are especially important in croplands. And at the local scale in experiments, we have found that unfortunately, croplands, because of the perturbance that we do to those soils, they have a very low capacity to fight pathogens. Sensibly, we have also demonstrated that by bringing back the microbiome, native microbiome, through microbial synthetic communities, through probiotics, we can actually help croplands to recover the capacity to fight against pathogens. So soil biodiversity is very important for ecosystem function, for soil function. Soil biodiversity very important for One Health. But soil biodiversity, soil organisms, they are very important for every single aspect of our life, and that includes as well the conservation of monuments. And I would like to show you a couple of examples, both of them in collaboration with a group in China. In the first example, we have been investigating the status of conservation of the China Great Wall. You all know China Great Wall, one of the biggest, magnificent monuments built by humankind. However, what you might not know is that China Great Wall was built with different types of materials. Some sections of the wall, they were built with rocks, and you might have taken a picture in those sections. However, other sections of the wall, very, very long sections of the wall, they were built with compacted soils. And compacted soils, like those in these pictures, they are highly vulnerable to soil erosion as any other type of soils, especially because many of these sections of the wall, they go through dryland ecosystems that, again, they are also very highly vulnerable to erosion. So when we aim to understand the status of conservation of these sections of the wall, we found that they are completely covered by these green organisms that you have in the picture. These green organisms are our friendly biokraft-forming mosses, that they are very, very important in drylands to protect our soil from erosion. They provide soil fertility, etc., etc. But we didn't know what they were doing in our wall. And what we found is that in sections of the wall that they are covered, these patches of the wall that they are covered by biokraft-forming mosses, they are far more resistant to erosion compared to patches that they are not covered by biokraft-forming mosses. So biokraft-forming mosses, these soil organisms, they are protecting the China Great Wall for the next generation. That's how important these organisms are. The other side of the coin would be the very well described, very well known effect of microorganisms on the biodeterioration of monuments. Monuments that many times are in our cities, that they are part of the urban microbiome of our cities. And that is what we found in this particular collaboration with a group in China. When we investigated this mausoleum almost 2,000 years ago, Eastern Han Dynasty, very important for the Chinese culture, where we investigated the mausoleum. The walls and the paints of the mausoleum are completely covered by these white organisms that they are actinobacteria, very well known to degrade this type of monuments, also to have huge capacity to degrade organic matter. What was very cool here is that when we investigated the origin of these actinobacteria, they are just a subset of the taxa living outside of the mausoleum. So pretty much soil here is a major source for these organisms that can have a huge impact on our monuments. So again, soil organisms, very important for conservation of monument, important for soil processes, important for One Health, but also I would like to highlight that soil organisms and ecosystem function, they are highly vulnerable to environmental stress, to global change. Global changes like climate change, the planet is getting warmer, some of the regions of the planet, they are getting huge aridification processes, but also I would not like to blame everything on climate change. Climate change is here. We need to adapt to climate change. We need to reduce carbon emissions, but also we have a major direct impact on our soil through land use intensification, a major impact that we need to control through processes such as, for example, deforestation, conversion from a native forest to plantations, cropland and grazing pasture, urbanization, 7 out of 10 people will be living in cities by 2050, growing human population, and also agriculture feeding this growing human population have major impact through pollution, erosion, etc., etc. So global change have major impacts on soil biodiversity and function, and I would like to show you a few examples from our own research. First example is coming from a decade of research in dryland ecosystem. At the global scale, regional scale, in experiments where we have been systematically finding that aridity have a negative effect on the biodiversity and on the function of our soils. Very cool, back in 2020, we found that actually this relationship between aridity and biodiversity and function is not always linear, but at certain levels of the aridity gradient, there are huge impacts from aridity on biodiversity. There are certain levels of this aridity gradient. There are these aridity thresholds, wherein small increases in aridity can have a very negative effect on biodiversity and function. Some of these aridity thresholds, they actually match with some emblematic transitions found in dryland ecosystems, such as, for example, the transition between semi-arid and arid ecosystem, wherein a small increase in aridity can have a drastic, non-linear negative effect on plant productivity, biodiversity, soil and plant biodiversity, increasing the prevalence of pathogens, decreasing the prevalence of beneficial organisms, or decreasing the carbon sequestration. So a more arid planet will not look very well in terms of soil biodiversity and function. Another major driver of climate change is warming. The planet is getting warmer. We are having a very tough summer and now fall in the south of Spain. And our own research have provided evidences at the global scale across natural ecosystems that warmer soils, they also have greater prevalence of pathogens such as Fusarium and Alternaria that can have devastating effect on our capacity to produce food, so on food security. Also, a warmer planet will have a lower prevalence of beneficial organisms such as fungal decomposers. As you know, fungal decomposers from this presentation, they are very important for the stability of the planet. Also, a warmer planet will have lower carbon sequestration, lower capacity to mitigate climate change, and many times also going through temperature threshold, wherein small increases in temperature can have very negative effect on carbon or the proportion of decomposers. So warmer planet, more pathogens, less beneficial organisms, less carbon. And I would like to keep this very simple pattern in mind because unfortunately it will keep ringing again and again during the rest of the presentation. Urban ecosystems, they are not that different from natural ecosystems. And in fact, across urban green spaces, city parks and gardens at the global scale, we have found that warmer city parks and gardens, they also have a lower prevalence of beneficial organisms such as ectomycorrhiza fungi. And also warmer city parks and gardens, they also have a greater prevalence of fungal plant pathogen, meaning ecosystems, city parks and gardens that will be more difficult to manage, less economically sustainable to manage in the future. So as you can see, climate change can have huge impacts on biodiversity and function, but I would not like to blame everything on climate change. Land use intensification have major effects in our soil. And I would like to put you a few examples from our own lab. First example is deforestation. I'm not sure if you're aware, but every year we lose 10 billion hectares of native forest, the equivalent to 14 billion soccer stadiums. We were very interested in understanding how this deforestation, transformation from a native forest to a plantation, a grassland, or a cropland, affects our soils. So we put together this global meta-analysis, and what we found was pretty straightforward, and you have already heard about that in this presentation. Deforestation reduced the amount of carbon of our soils, increased the prevalence of pathogens, and decreased the prevalence of beneficial organisms such as ectomycorrhiza fungi. Another major driver of global change, our start topic today, is urbanization. 7 out of 10 people will live in cities by 2050. Urban green spaces, the transformation from a natural ecosystem to a city park to a garden will have critical effects on the biodiversity and function of our soil. However, we didn't know much about this a few years ago. Urban green spaces, they are fundamental for our life in the future and also today for our mental health, for our physical health, and also for regulating flooding events or mitigating climate change in our cities. So understanding how these urban green spaces affect our soil, how they behave compared to natural ecosystems is something that is very important for us. And with that idea in mind, we put together this survey that I have introduced before, comparing this nearby natural ecosystem with urban green spaces, city parks and gardens, and where we have found major positive and negative effects of urban green spaces compared to natural ecosystem on the biodiversity and the function of our ecosystem. And I would like to show some of these results with you today. For example, we have found that urban green spaces, they normally have a lower plant biodiversity compared to nearby natural ecosystem, a lower number of species in terms of plant diversity compared to nearby natural ecosystem. But very interestingly, the opposite pattern is found for some of the most dominant organisms of the planet, for the diversity of bacteria, where urban green spaces, they actually have a positive effect on the diversity of bacteria, and also the diversity of protists, and even the diversity of functional genes at the global scale. You will see during the rest of the presentation that some of these effects are actually related with, for example, the perturbance of pH, for example, the effect that we have on urban ecosystems, right? So, for example, you will see in the next slide how perturbance, the impact of humans, of city parks and gardens, increase the pH of the soil, and soil pH is the major driver of soil biodiversity. And because of that, we believe that the biodiversity of bacteria, biodiversity of protists, and also functional genes is actually higher in city parks compared to natural ecosystems. Very cool too. Unfortunately, we also found that urban green spaces, city parks and gardens, they actually have a huge impact on the biotic homogenization of our soils. They promote microbiomes that are very similar across city parks and gardens at the global scale, especially when you compare it to the huge heterogeneity that you have for natural ecosystems. It is very simple to explain, right? You think about city parks and gardens in New York, Madrid, Barcelona, they have very similar type of vegetation, very similar type of management. And also they are very different from natural ecosystems in the way that we manage these environments. And because of that, we end up selecting a very similar microbiome across the globe and end up having a huge homogenization effect on the soil microbial community, which is something that has been also observed for other biotic communities, for birds in other studies. This is exactly also what we found in urban green spaces across China in this study that we published this year, where we compared 4 land uses, native forests, croplands, city parks, residential areas across China, and where we found exactly the same, that city parks, they have a huge homogenization effect on the microbiome of our soil. So the city parks and gardens across China, they look very similar to each other. But very cool in this study, something that we haven't observed before, was that the homogenization effect on the soil microbiome was even higher than that found in agricultural lands. And agricultural lands, you will all know, they have a huge homogenization effect on plant and microbial communities. So urbanization can have a huge homogenization effect on our soil organisms by selecting a particular microbiome because of the conditions that we create in the city park or garden, like this increases in the pH, because of the perturbance that we have in our soils, because we reduce the organic matter, because of the erosion that we create. This higher pH promote a particular microbiome and greater biodiversity. In this global study, we found, for example, that there are 500 taxa of bacteria, fungi, and protists that they are actually found across city parks and gardens at the global scale, the equivalent to pigeons, squirrels, and also pounded trees that you can also find across city parks and gardens worldwide. The only difference is that, right, you don't see these microorganisms that they are the unseen majority living in our city parks and gardens. Very cool. Again, when you have a higher pH associated with these human impacts, you actually have a greater prevalence of this urban microbiome. Also associated with this particular issue, with the pH of the soil, we have found that urban activities, they can promote the range of expansion of like the biodiversity of soil protists from temperate ecosystem to subtropical and tropical forests. I'm not sure if you're aware, but tropical and subtropical soils, they have very low pH. As I have been trying to explain, pH is the major driver of soil biodiversity. So pH is a major environmental filter in tropical ecosystems for many soil microbial communities. However, by creating city parks and gardens in urban green spaces in tropical ecosystems, we increase the pH, and that opens the niche for organisms from temperate ecosystems that shouldn't be there, creating an expansion of the microbiome at the scale of China and also at the global scale of organisms that they shouldn't be there, that they are invading these ecosystems. So again, urban green space is very important for the biodiversity of our soils, and also urban green spaces are very important for ecosystem function, for the multifunctionality of terrestrial ecosystems. Something that we traditionally thought is that city parks and gardens were relatively perturbed ecosystems, that they were different from natural ecosystems, and they look different from natural ecosystems. However, in terms of ecosystem function, in terms of multifunctionality, city parks and gardens, they are actually very similar to natural ecosystems. In this global collaborative network, we found, for example, that the multifunctionality of city parks, gardens, and natural ecosystems, they are very similar in terms of soil stability, water regulation, plant productivity, decomposition. They actually look exactly the same. They also look exactly the same in terms of carbon sequestration. where urban green spaces and nearby natural ecosystem, they have similar capacity to sequester carbon in our soil, and therefore similar capacity to mitigate climate change. And this is very good news because it actually suggests that our urban green spaces, they are a very nice sink for carbon sequestration. So very, very, very cool news. Also, urban green spaces, they behave very similar to natural ecosystem. That it was also a surprise in the ways that, as you know, natural ecosystem, they take time to develop. They go through successional processes wherein their capacity to function increase with time. However, we didn't have that sort of information back in the days for the soil organisms living in our city parks and gardens. And in 2016, we put together this study in Eastern Australia, where we compare 3 different settlements that they have very different ages. Richmond is more than 200 years old. But however, Jordan Spring, for example, is a very new settlement, and their city parks and gardens, they have very different age. And what we found was that age is very important in explaining the function of city parks and gardens, where older city parks and gardens also have higher capacity, for example, to decompose organic matter. So very cool, this ecosystem, they seems to behave very similarly to natural ecosystem. Unfortunately, as till this point of the presentation, we have been talking a lot about city park and garden, but unfortunately, not every city park and garden, not every urban green space is exactly managed in the same way. You know that there are many different urban green spaces, from more natural urban green spaces like city parks that I have been showing you some results today, but also you have a different bunch of urban green spaces like roundabout, golf courses, urban farms, when you have a higher intensification. Land uses have higher intensification in this particular urban green spaces. So a few years ago, we were very interested in understanding how land use intensification influenced biodiversity and function in urban green spaces. So we applied for this project, the Urban Change Project, and conducted this network of collaboration in the Iberian Peninsula, where we surveyed 51 cities and we surveyed soils and these 5 types of urban green spaces, from less managed natural and city park to more managed, a priori, more managed golf course and urban farms. And what we found was something pretty straightforward. Land use intensification decouples the biodiversity and function across urban green spaces around the Iberian Peninsula. If you take a look to this slide, the x-axis have the different land uses that we survey: natural ecosystems, city parks, roundabout, golf course, and urban farms. From left to right, intensification. And then in panel A, you have multifunctionality in the y-axis. Panel B, you have biodiversity in the y-axis, the biodiversity of plants, number of species of plant, but also number of species of bacteria, fungi, protists, and invertebrates. And what we found is that land use intensification decouples biodiversity and function across the Iberian Peninsula. So you have less ecosystem function with intensification, however, you have higher biodiversity, number of species with intensification associated with this pH effect with the way that we perturb our soil. So this information for this paper that is now under review is very important if you want to develop new urban green spaces in your cities. Also, another example of global change or major changes in urban green spaces is the case of pollution. Our own research at the global scale have provided solid evidences that urban green spaces, they accumulate a lot of pollutant, heavy metals, pesticides, microplastic, antibiotic-resistant genes. But also very alarming in this study, we found that nearby natural ecosystems, they actually have similar levels of pollution for many of these pollutants. So it's very important that we understand how to conserve our urban green spaces, our natural ecosystem from this pollution coming from our cities. So more resources, more research need to be done in this direction. So as you can see, urbanization have a huge impact on biodiversity and function, sometimes positive effects, sometimes negative effects. But I would like you to go back now to the bigger picture where global change, climate change, land intensification impacts the biodiversity and the function of our soil. And if you think about it, in this particular presentation today, we have been so far talking about individual drivers, how warming, how acidification, deforestation influence the biodiversity and the function of our soil. However, unfortunately, soils, they are not subject to a single stressor at the time. Soils are subject to multiple global chain stressors simultaneously, and understanding how global change, multiple global chain stressor influence our soil is a major priority. With that idea in mind, a few years ago, comparing two independent global surveys, we investigated how a number of stressors above a high level of stress, and by stressors here I mean aridity, high temperature, seasonality, salinity, changes in the chemistry of the soil, heavy metal, human impacts, all these factors that we have been considered individually, they can affect the biodiversity and the function of the soil. And what we found was pretty straightforward, kind of expected. If you increase the number of stressors over a high level of stress, you have lower biodiversity and lower ecosystem function. So very important that we understand how to protect, how to conserve our soil, because soil biodiversity is critical for function, but highly vulnerable to global change. And this is one of the major goals of the Biofarm Lab, to provide innovative knowledge, to provide solutions for land managers, for policymakers, to have the possibility to take informed decisions. Tools such as, for example, the first global maps for the distribution of bacteria, the dominant bacteria in soil in 2018, dominant fungi in soil in 2019, or the distribution of soil mosses that you have already know that they are very important in 2023. This type of maps, if you think about it, they have existed for emblematic organisms such as the polar bear or the distribution of plants for decades or even centuries. However, we did not have this sort of information until recently. And these sort of maps, they are very, very important to understand the global hotspots of soil biodiversity and ecosystem function. That is what we tried to put together back in 2022 when we published this study, aiming to understand the global hotspots for biodiversity and function that you can see in dark brown in this particular image. Unfortunately, in this study, we also found that these global hotspots for biodiversity and function, they are highly unprotected. They don't overlap with natural parks, with national parks. Less than 10% of these hotspots, they are protected today. So this is very important to understand how conservation is affecting these global hotspots of biodiversity and function. Even more alarming, I'm not sure if you're aware, but we don't really even know if protected areas are actually protecting our soils today, because protected areas, natural park, national park, they were developed to protect our emblematic organisms such as the Iberian lynx in the Doñana National Park or other plants or geographical accidents. However, soil conservation wasn't really taken into account in most cases. So we don't really know if national parks, they're actually protecting our soil. So with that idea in mind, we got this data, which is available online from the Lucas survey. I'm not sure if you're aware about this survey, but the Joint Research Center from the European Commission is conducting a monitoring of soils in the European Union with more than 800 sites. And this data is from 2018, from the first point of their survey. Using this data, we compare protected and unprotected ecosystems and the biodiversity and function, the different parameters that you have in this slide, and found something pretty straightforward. Natural parks, these national parks in general, they don't seem to have different levels of biodiversity and function than unprotected ecosystems. And at the least of the cases, this uncertainty is not something very good for the biodiversity and conservation of our soil. So we really need to learn more about how these natural parks, national parks, are protecting our soils. With that, I would like to start wrapping up this presentation with some take-home messages. The first take-home message is that protecting our soils is critical for the biodiversity and the function of croplands for producing food, but also the conservation of soil should go beyond food production, and we should care about the overall soil biodiversity, the largest portion of the biodiversity of the planet. We shouldn't forget that. Also, for the sequestration of carbon, the largest stock of soil carbon, organic carbon there in our soil. For One Health, for the conservation of monument, we should care about the conservation of our soils. And again, I believe that we should be able to do much more than what we are doing these days. I show you in these slides that land use intensification have a critical negative effect on the biodiversity and function of our soil. However, sustainable management can also have a very positive effect on our soil, on our capacity to recover our soils. And I would like to show you a couple of examples from our lab. The first example is this meta-analysis was conducted by one of the PhD students in the lab, Luna Medrano, where we investigated how sustainable agriculture can promote biodiversity and function in cereal fields. Wheat, maize, some of the major staple foods for our food security. And what we found, something pretty straightforward, if you use rotations, if you have limited tillage, or you have crop residue, that can have a very positive effect on soil fertility, on carbon sequestration, on organic matter decomposition, without having a very negative effect on the production of food, or even having a positive effect if you use crop residue, which is information that is critical for the farmers in agricultural lands, but also in our urban farms ecosystems. So sustainable management is very important. Very similarly, in natural ecosystems, restoration, rewilding is something that is very important too. Not sure if you're aware, but we are in the United Nations decades of ecosystem restoration, 21, 2030. And a few years ago, we were very interested in understanding how rewilding, the restoration of ecosystems, especially in forest ecosystems, influence the biodiversity and function of our soil. And what we found in a global meta-analysis was that with time, actually, you can bring back plant biodiversity, plant biomass, microbial habitat, soil carbon, soil fertility, etc., etc. So restoration, management is very important for our ecosystems and for bringing the health of our ecosystems back. With that, I would like to leave you with a last take-home message, this sentence this quote I like from President Roosevelt from 1937. The president of the US, he was targeting his governors in the different states in the US, making a very simple message. The nation that destroyed soil, destroyed itself. That's how important that message is today for us. It was 100 years ago. So let's make soil great again. Let's convert something bad, let's transform something bad into something good. So with that, I would like to thank you for your attention. And of course, I will be happy to take any question. Thank you.
Well, it was an impressive presentation. I really like. I don't know if we an applause here, but it was super good. I would like to open the floor for questions. If not, I have some, but there's a lot of people here, more than 20 people today, so don't be shy. Connect the microphones and we have an expert today here, so let's use these 15 minutes we have to speak a little about urban ecology or all the soil results we have here today. Any questions in the room? The chat, maybe? Well, I can start while people decide whether they wanted to ask or not. For some of the results you presented today, I have seen that the age of the city was a super important factor for the functioning, I think, of the biodiversity of the city. Do you think the most important driver of change in this case will be the establishment of the city, so the land use change in this moment, more than how the city developed after that? So the urbanization per se, the construction of the city has more impact, and then the ecosystems try to recover until the pollutants or climate or different climate, but those impacts are not that important as the primary change.
I think that everything happened in parallel and probably depend on the particular biodiversity or function that you are interested in. So it might be different for different aspects. But in the case, for example, of soil biodiversity, as I was explaining in the presentation, some soil properties like the pH of the soil, they are very important. And as time passes, these properties, they are known to change ecosystems, they are known to accumulate more organic matter. There are major changes that they can affect the biodiversity and the function of the soil in terms of, for example, the accumulation of litter for organic matter decomposition. So a very new ecosystem, a city park that is just built today, might still need time to actually recover from the perturbance, but also like in a secondary succession, start accumulating organic matter, litter, so that it can actually support all these ecosystem services that many times in our soil they are related with the organic matter decomposition. But of course, changes in the policies, the management types in these environments, they can actually have a restarting effect certain levels, right? Because certain ecosystems, especially gardens, they change plan constantly, so that wouldn't be exactly the same that in a city park with, like in an urban forest that actually it allows a better development with time. So
yeah. Thanks. Related to this, to the species composition turnover and everything, and then I let Gema asking. Sorry. How do you think the contribution of the exotic species? Because well, in cities we could expect higher number of exotic than in natural areas, and in some of your results you have seen that there is a increase in diversity in some places, but there is not an increase in multifunctionality. No, there's this decouple. And do you think this could be due to the exotic species we could have in cities?
not necessarily, although again it would depends on the type of organism, right? But for example, if we talk about plant species, what we have found is that in urban farms and certain type of more managed type of ecosystem, you might end up having like a lot of species, but with type of species, right? So not necessarily need to be the majority of the community. What I was showing, for example, was the number of species, right? So there are more presence of different weeds than in other type of ecosystem. But yeah, exotic species are very important in this type of environment because especially in gardens, again, we plant a lot of like exotic ornamental species, right? That they shouldn't be there. So these type of ecosystems are very prone to this type of species. And also very cool what we have found with microbiomes, that it is something not so well established, the microbiome invasions and so on, that they haven't been so well established because of the limitations in the technology that we use for sequencing and so on. And something very cool that we have found recently, this paper in PNAs that we published this year in collaboration with a group in China, is that actually by creating this type of environment, city parks and gardens and subtropical ecosystem where certain species, they shouldn't be there, we open niche for those species because we increase the pH of the soil, so that environmental filter that is the low pH disappear, and all of a sudden temperate taxa that they shouldn't be in a tropical ecosystem, they start living in our tropical ecosystem. So that's something pretty cool from the point of view of invasion, but not so well known as in the case of plant ecology or animal ecology, for example.
Very cool. Gema, you are waiting for a...
Hi, can you listen me?
Yeah.
Okay, because today I have these days at the lab when everything is crashing. First, thank you very much. It has been simply impressive. And I have a question because well, I'm working with ants in urban areas and I'm using them as bioindicators. And what I found when in the times that I had a chance to repeat the samplings in different cities in different countries, it's like I found a high variability in the results depending on the country, mostly because of the management. In your studies, you have shown general patterns which are pretty similar to the patterns I found with the ant biodiversity or functional traits. But I would like to know if also in the case of the soil, in terms of biochemistry, but also the biodiversity, did you find such a large variability among countries? Because in my case, it's like, for example, comparing Poland with Germany, which are pretty similar weather and so on, or Poland with Spain. But in your case, you are having much larger variety of habitats and so on. Did you find this large variability at the local and the country scale, let's say, larger scale?
Yeah, thank you, Gema. That's a very good question. Yeah, we found a huge variability. I mean, if you think about climate or soil properties, you have like all sort of like urban green spaces, city parks and gardens. They can be everywhere, right? So they can be in a dryland ecosystem, in a more basic ecosystem, in a tropical ecosystem. So you end up having like very different type of ecosystems, a lot of variability, but also we found a clear signal in terms of how soil property move in this type of environment. For example, the increase of the pH in one of these management signatures that we found in these ecosystems. And in general, we see patterns, but of course, with a lot of variability. Also, I guess that in the case of ant, that is something very cool. The species that you have in different places might also differ a lot, but in terms of the microbiomes, especially bacterial communities, many times they move easily, right, at the global scale, because many times they move through the air and spores and et cetera. So the way that these organisms behave at the global scale is different to animals or plants that they are more locally identified, right? So in that way, even when we found a huge variability, we also find a very clear signal. For example, in the case that urban green spaces have greater number of species of bacteria or protists compared to natural ecosystems in our soils, and we found that in that global survey that we conducted a while ago, but also in this more recent survey that we conducted in the Iberian Peninsula. So it seems to be quite consistent pattern, but might depend on the organism, right? Might be different for birds, might be different for ants, etc.
And I have a second question, because, for example, at local scale, and it's a bit connected with Juan Antonio's previous question, we have been also checking the biodiversity in the areas invaded with goldenrod. And the thing is, like, when we had these areas invaded with goldenrod, we found different micro habitats in terms of like there were some spots within the same park or the same meadow in which where you have the Solidago species, they are much warmer and much drier than the other spots. And our biodiversity was fully different. The anthropofauna in general differ a lot depending on the spot you were measuring within the same meadow, only because of this invasion. So in your case, now that you have been talking about the bacteria and so on, the dispersal of this organism, I believe that is also important if they can establish. Is it also happening, or like the species, the number of species that you have are mostly generalist, so they do not have such big troubles in establishing in this different microhabitat within the same meadow, for example?
Yeah, that's a good point. We have generalists and specialists. That's not an issue. You need to think that the scale for plant or animal ecology is completely different than for soil organisms. You need to think that as I was mentioning in the presentation, a gram of soil already contains thousands of species of bacteria and millions of individuals. So it's slightly different in the way that we kind of account for the spatial heterogeneity is that when we conducted our surveys, we work with composite samples, so that we work. Like imagine like in a 30-meter plot, and then we collected a bunch of soil sample, and then we put them together, work on composite samples so that we can actually capture the heterogeneity. In general, soil bacteria, protists are highly associated with soil properties of the soil, and these properties at this scale, they are relatively homogeneous in a plot, especially like in a disturbed type of ecosystem. So, soil pH would be relatively similar, and therefore you will have very similar type of communities at that scale. But also you will have a bunch of different specialists and so on that might be related with some species of plants that when that species of plant is not there, might not be there, etc. So in general, we try to capture this heterogeneity in this way, but yeah, surely it will be different if you're looking for a plant or a bird, where that would be microsite might be far more important that way.
Okay, thank you very much. And again, congratulations for the amazing job.
Thank you.
Javier has a question. Javier Cano.
Yes. Hello. Hi, Manu. How are you?
Hey, Javi, how are you?
Not too bad.
Yeah, cool to see you, man.
It is. Long time ago. So, yeah, well, I would like to join to all the congratulations and, well, trying to follow your presentation. I had some questions with the role of pH, because apparently it's one of the most important factors in soil to determine the biodiversity. But I think looking at your studies, in some of them, because the pH increases in the urban parks, no?
Mm.
And that will lead to higher level of biodiversity. But seems that overall, the biodiversity in urban areas is lower than in natural areas. Or is this...
It's not lower, it's more homogeneous. So you need to, there are different aspects in biodiversity, right, that you need to consider. Like we work with alpha diversity, number of species, we work with beta diversity, right? Whether that diversity is similar across sites or different across sites, right? We work with gamma diversity. So what happens in urban green spaces is that you have a greater number of species of bacteria that happen in places that have high pH, that happen everywhere in natural ecosystems too. But those species are actually very similar across urban green spaces. So if you put all those urban green spaces together, you end up having a lower biodiversity compared to the heterogeneity of natural ecosystems. But if you count those species in the city park or garden, you have a larger number of species, right?
Okay. And related to this question, do you think that adding limestone to the soils will increase the level of biodiversity, or is not related to?
It would depend on the ecosystem that you're working, Javi, because and why you're doing that, right? Like if you are in a soil with a very low pH, you will end up having like lower number of species, but also those species, they actually might be very different to the species that you have in another place with low pH. While in places that have high pH, we have found over the time in natural ecosystem, urban ecosystem, that you end up selecting for similar species that like high pH. You have more species, but they are also similar. So it depends on what you are actually targeting. It might make sense to do limestone or not, right? Because you might also affect other soil processes and function that you might not want to affect, right?
So in terms of rewilding, what will be your advice here? Just limit the human intervention and let the nature follow its way, or what is your idea about this?
It depends on the ecosystem. So you are in a natural ecosystems, the approach that you will take will be different than in a cropland or a urban ecosystem, right? Like in a city park or garden, of course, you need to have management, right? Because at the end of the day, this type of environments, they are actually managed so that we can take advantage of the ecosystem services that they provide, right? So that we can go there with our pets, our dogs, so that we can go there to play soccer or whatever, basketball or whatever we are doing, right? So they need to have like a management, right? So it could be about, implementing management that they are sustainable and that they can actually, somehow interact with these ecosystem services to get the best out of it.
Okay, thank you so much.
Yeah, thank you. Great to see you. Bye. Hopefully see you soon somewhere.
Yeah. We need to meet. Okay, so yeah, thank you. No more questions.
Jessica, you have a question for Manuel?
Hi, congratulations. Thank you for accepting the invitation to come here and talk about your work, because it's just amazing. So thank you for starting all the seminars.
Thank you.
I have a question that maybe is like difficult to answer. For example, regarding the agricultural land, that they are very they are very stressed because they add a lot of pesticides, a lot of fertilizers that change a lot the soil. If suddenly we change the way in where we manage and we start doing like practice, as you mentioned at the end of the presentation, like we have an idea of how much long it will take to turn this soil into like more effective or functional soil, like more natural or with the functions back?
Yeah, that's a good question. I don't think we have necessarily a very good idea about how long it will take, and it probably depends on the climate where you are, right? Like it's not the same to be in a very highly productive type of ecosystem compared to like a cold or a very dry ecosystem where the processes they happen to be slow. That's something that we know for natural succession, for example, and even for long-term ecosystem development. So it will really depend on that, but also, it will really take a while into getting to like an equilibrium of the ecosystem where this makes sense, right? Like it is like a controversial topic in the way that when you move from a conventional agriculture where you have intensive management, where you produce a lot of food, not necessarily the greatest quality, but you can actually produce more food, convincing the farmer to move to a sustainable agriculture, especially because there are so many different aspects like ecological agriculture or like organic agriculture, and not necessarily they are exactly the same management practices. Sometimes it's just about changing fertilizer, mineral fertilizer for organic fertilization. Sometimes it's about stop tilling or doing a less aggressive type of tilling. Sometimes it's about rotation. So implementing these management practices, they definitely are very important if we want to conserve our soil, if we want to bring back carbon sequestration, we want to bring back fertility, we want to bring back organic matter decomposition, but also need to be done, of course, always not missing the important point of this ecosystem that is producing food, so that we can actually engage the farmers and actually try to do this all together. But yeah, our own research, this study that is now under review, this meta-analysis led by Luna, she found that sustainable agriculture actually have a very positive effect on all these different aspects from our soil, and actually doesn't really have a negative effect on cereal production. So why not to go for it?
Okay, thank you so much. So interesting, the answer.
Okay, I think we don't have any more questions, and maybe we are more or less on time of the hour. So I will like to thank Manuel for being here again. So thanks a lot for your help with these seminars, and we will have another in 1 month. I would like to share the announcement of the next seminar. Is gonna be given by Gema Trigos Peral. She was here. I don't know if she's still here, asking some questions. So in 1 month, we will speak about the ant communities she presented today. So with this, so we can finish today. Thanks everyone for being here. We will have the recording in YouTube in which you want to revisit this, because it has been super interesting and like super complete. So thank you, Manuel. Thank you everyone for being here, and see you in 3-4 weeks.
Thank
Thank
you.
you.
All the best with the seminars. Bye bye.
Thank you for the
Bye.
presentation.